talk a little bit about, I kind of coined the term ag-environ research, which is basically looking at the impact of environmental practice, agricultural practices on our environment. I like to use this slide when I'm talking with my class about the earth is shrinking, which means it's not really shrinking physically, but in terms of the space that's allocated for crop production and crop growth, just based on human population growth and the land that's actually available, is in fact getting smaller. And what that means is while we have to feed more and more people with limited resources, we have to look at this balancing act. Here's an example that way back in 1930, one farmer would have to be expected to feed like about 10 people. In about 1970, that went to 73 people, and now it's like about 150. And what does that mean? It means we have to produce more and more food and less land with less and less resources. So yes, we're trying to optimize our overall productivity, but when it comes to an environmental aspect, do our agricultural practices and processes provide safe, healthy food that a lot of people are starting to address? Secondly, what about our agricultural production systems on our natural resources and overall environmental quality? So our ag-environment research component that we've termed broadly, that involves a number of people that you'll be see talking with both the plant science department and the other interactions that we've had, is how does this agricultural practices influence our water, air, and soil resources? And my component, I'm trying to focus primarily on a nitrogen component, because you can't really look at everything, and they're looking at nitrogen fertilizer. So the three examples I'm going to show you is one that's related to air, water, and then finally soil. And soil is the one that we've just recently going to be working together with Dr. Adisham. When it comes to air quality concerns, we have the greenhouse gases and the impact of those. One of the most potent one is nitrous oxide. Nitrous oxide is like 390 times more potent as a greenhouse gas than CO2. And the EPA has its established method of looking and trying to quantify these nitrous oxide emissions from agricultural practices. Our work to date has been primarily looking at different fertilizer types and rates. And what that brings to the table here for us is, in addition to getting, I'm already excited about some of the work that I've heard from the previous speakers, and I know we can have a little more interaction, or more interaction as we move and hear the other speakers, is field work and sites that can actually be available for collaborative work, as well as some of the instrumentation. And I'm going to leave that later on for when Sharon introduces some of there's a one-page summary of the kind of instrumentation that we have here. So we can have opportunity for collaborative work both in the field and in our instrumentation here. This is an example of where we're using these chambers to try to determine the nitrous oxide fluxes from some of the different fields that are subjected. That's part of the air quality. And then, since I'm all tied up with the nitrogen cycle, you might be addressing one component of the nitrogen cycle and either adversely impacting on the other component. So nitrate leaching is where we look at the potential for nitrate going into our groundwater system. And where we look at here is looking at nitrate, potential for nitrate leaching by looking at different cropping systems, and primarily since my area is a vegetable crop, I tend to focus on that. We're looking at the impact of comparing different fertilizer types and fertilizer rate and the potential for nitrate leaching. That involves a bunch of work that's agronomic practices, where we move from planting stuff in the plants out there, fertilization practices, sampling. So it's a combination of field, lab, and collaborative work with growers. So there's a next bunch of opportunities. And then the one that I'm actually kind of like been spending most of my time with is looking at the option of aerating the water that's actually going to the root zone. And the work has been done so far that shows that yield is an issue. It's not an issue. We've knocked that problem off. So the next one we're trying to look is try to understand what this actually has in our soil microbial component. And this is where Dr. Dijon and we just got into. We're also looking at salinity, but we are seeing that there's a difference in the microbial population as we look at the DNA. So it's kind of exciting here to be able to have this opportunity to talk to a group of people. And I can see that this just expanding from our basic philosophy, where to have a success in all this research, we have an excellent research team. We like to call them, this was the grad lab, the team of 2017. We kind of coined that term after some of their performance in bowling, but it just goes to show that we can extend this with almost everybody who is in here, in one way or the other. So with that, we're open for collaboration, and a lot more will become available when the other speakers. Okay, thank you.